Hello students, the topic is molecular basis of inheritance, and in today's class let us understand the concept of the genetic code. Students, you have learned about the central dogma of molecular biology, which says that DNA gives RNA and RNA gives the proteins. So, it is the RNA which is responsible for giving the proteins, which in turn are sequences of amino acids. Students, you will notice that RNA comprises of nitrogenous bases and proteins are made up of amino acids. It is very easy to get a RNA from DNA because DNA and RNA both comprise of nitrogenous bases. Thus, through the rule of complementarity, from a DNA, a RNA can be easily obtained by the process of transcription. But when amino acids, which are completely different from the nitrogenous bases, have to be created, the process becomes more complex. It was George Gamow, a physicist, who argued that as there are only 4 types of nitrogenous bases in RNA, that is adenine, guanine, cytosine and uracil, and as there are only 20 types of amino acids in all living cells from the prokaryotes to the eukaryotes, there should be some combination of the nitrogenous bases which represent the amino acids in the cytosol. Thus, they tried to have various permutations and combinations to see how many nitrogenous bases could be assembled together so that they represent an amino acid. First, when they thought of a codon with only 2 nitrogenous bases, 4 into 4 would give a permutation and combination of 16 codes. Now, 16 codes were insufficient to represent 20 amino acids. Thus, they thought of a code comprising of 3 nitrogenous bases or combination of any 3 nitrogenous bases among the 4 types, that is adenine, guanine, cytosine and uracil. Thus, 4 into 4 into 4 would give 64. Now, 64 codes are too many to represent 20 amino acids. However, it is possible to have this combination rather than having 16 codes which represent 20. Thus, it was a very bold proposition based on permutation and combination made by the scientist George Gamow, who said that there should be 64 codons which represent the 20 amino acids. This information was used by the Indian scientist and Nobel laureate Hargobind Khurana. Hargobind Khurana made use of Marshall-Nirenberg's cell-free system for synthesizing proteins from RNA. You know, students, that inside the cell, the RNA is used for synthesis of proteins, and this happens on the ribosomes. However, the Marshall-Nirenberg cell-free system was capable of producing these proteins in an artificial medium outside the body. Thus, Hargobind Khurana made use of this system to prove that the RNA comprises of a codon which is made up of triplets, which was responsible for bringing the amino acids in the desired sequence to generate a protein. So let us understand the salient features of the genetic code. Thus, it was understood that the codon is a triplet comprising of 64 codons. Out of the 64 codons, 61 codons code for amino acids and 3 codons do not code for any amino acids, hence they are called as the stop codons. You can see that, students, the stop codons don't represent any amino acids. One codon codes for only one amino acid. Hence, the codon is said to be unambiguous and specific. That means for a particular codon, only that representative amino acid will come to the ribosome for the purpose of protein synthesis. Thus, this nature of the codon is called as unambiguous. The codon is also said to be degenerate. What we mean by degenerate is that there could be more than one codons representing an amino acid. Thus, the 64 codons what we mentioned also tell us that each amino acid could be represented by more than one codon, and this is called as the degenerate nature. The codon on the mRNA is read in a continuous manner with no punctuations. The codon is nearly universal. That means all organisms which have cellular nature have a same system of producing proteins from the mRNA, and thus we say that the codon is universal or the genetic code is universal. However, very few changes may be seen in some bacteria, but otherwise the codon is completely universal. In all organisms, it is seen that AUG always comes as the first amino acid and represents the first amino acid, and hence it is called as the initiator codon. Now, this initiator codon has two main functions. That is, when it comes in any portion of the mRNA which is not at the beginning, then it represents the amino acid methionine, and when it is in the first position on the mRNA, it is called as the initiator codon. Thus, AUG represents methionine in either case, whether it is in the beginning of the mRNA or whether it is in the middle portion of the mRNA. However, in the absence of AUG, the process of translation cannot begin, and hence it is called as the initiator codon. So let us just look at the structure of the genetic code which was deciphered by Hargobind Khorana. So you can see here, students, that the codon comprises of 3 nitrogenous bases. For example, UUU and UUC represent phenylalanine. UUA and UUG represent leucine. And likewise, you can see that every amino acid here is represented by a triplet. And thus we say that the genetic code is a triplet. We can see that AUG is the code which is representing methionine, and it is always present in the first part of the mRNA and therefore called as the initiator codon. We already mentioned about the stop codon. So you can see, students, that UAA, UAG and UGA are called as the stop codons, and the stop codons do not code for any amino acids, and the presence of these codons therefore will bring an end to the process of translation. Thus, we can see that the genetic code disappeared, wherein it was said that it is made up of 3 nitrogenous bases, and this genetic code is very much important in the process of translation, which is giving rise to the final product of the DNA, that is proteins. Thank you, students.